Hello, everyone, and welcome to today's webinar, Remote Autonomous Molecular Analytical Laboratories at Sea and on Land. I'm Susie Valdez of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by MicroGEM. To learn more about MicroGEM, visit them at microgembio.com. So let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing this presentation, just simply click on the Support tab found at the top right corner of your presentation window, or report the problem by clicking on the Ask a Question located at the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of your presentation window and follow the process to obtain those credits. I now present today's speaker, Christina Preston, a research specialist at the Monterey Bay Aquarium Research Institute. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Christina, you may now begin your presentation. Welcome.
Well, thank you very much for that kind introduction, and I hope that you enjoy my talk today. Well, today I'm going to talk about the instruments that are featured within these videos. And these instruments are our underwater laboratories. The one on the right shows our mobile platform known as a long-range underwater autonomous vehicle carries our sampling robot. And you just saw it being deployed, and now you're seeing the water column from the vehicle's perspective as it flies through the water column, and eventually it'll encounter a jam of jellies. The video on the left shows a person diving on a moored instrument, and both of these instruments are able to provide data while in their environment and are the focus of today's talk. And before I really get started, I wanted to at least talk a little bit about our group. I work at the Monterey Bay Aquarium Research Institute, and I work in a group that develops sensors in order to help facilitate underwater research. And we're a really diverse group. We span scientists that are microbiologists and molecular biologists, as well as engineers of mechanical, electrical, and software. So we're a really diverse group, and our main goal is to basically be able to provide new means to study in aquatic environments. And so I want to start introducing you to why someone would want to utilize robotic samplers, and then talk about really the challenges of moving the lab, meaning a molecular lab, directly into the aquatic environment. And I'm going to utilize MBARI's solution, which is known as the Environmental Sample Processor, and you might hear me refer to it as the ESP. And the idea of the ESP originated with Chris Scholin over 25 years ago, and Chris really wanted to move the lab into the ocean for the real-time detection of harmful algal blooms in our oceans. And although the ESP was originally designed for that one particular task, it's really expanded to a wide range of organisms and analytes. And you might hear me also utilize the terms second generation and third generation, or 2G ESP or 3G ESP, and that just refers to the version of that particular instrument. In today's talk, I'm really going to focus on reagent selection and testing that was performed to support the molecular assays for nucleic acid and toxin detection and quantification. The other capability of the instrument, sample preservation, has also been important for our various collaborators, as many of the methods that they use aren't quite amenable to automation by the ESP. This next video shows one of our ships leaving port and heading out on a research cruise, and MBARI is located just at the top of this particular, up the top of that video. And this is just to remind everybody that ocean sciences is quite expeditionary. It means moving your lab onto a ship and heading out to sea in order to collect the samples. And one issue of having to rely on ships means that you're also tied to its schedule, and one that's often determined a year in advance. And those schedules really have impacts as to when we can get into the field to study particular phenomena. We know in general the time of the year each particular phenomena occurs. For example, in Monterey Bay, harmful algal blooms occur in the spring phytoplankton and fall phytoplankton blooms. And so we know in general when they occur, but if you had to pick a week, maybe that bloom didn't quite get initiated then. So being able to leave behind an instrument that can span that time frame of a particular phenomenon that you want to study is a really great asset. It could monitor for, and once that phenomena arrives, you could increase its sampling rate and capture samples during that particular time. Often whenever we go to sea, we are trying to collect samples, and for many of us, that means bringing water back to the ship. This is one of those instruments. This is a CTD rosette with a bunch of water sampling niskins. And when these niskins go down, they're open like this example here, and they're open on both ends. So when you reach a depth of water that you want to sample, those niskin bottles close, capturing that water, and that's what you're seeing here, that those niskins have all been sampled. And then you bring that niskin bottle up to the surface of the ship. You have to drain the water, then you take it into the lab where that water gets filtered. So already you could imagine that you've collected the water. There's a delay between when it gets onto the ship to where you even get to drain the bottles and process. And there's also an environmental change. And so being able to process water in its environment is a really key feature. And a lot of people have started to develop robotic samplers that will actually do just that. They provide a way to either maintain a longer presence. They also decouple sampling from ship operations during a particular research cruise. And they also provide a means to preserve water immediately and under its environmental conditions. Some of these instruments also can perform experiments. They can either add tracers and then track that over time, or they can actually even provide molecular information. And if you're interested in these types of instruments, there's two really good reviews, one by Ottoson and one by McQuillan and Robidart, that are available. One thing that everybody's going after really are organisms that are captured within this droplet of water. And so really the ocean is a genetic soup of its resident species. And this is a great quote from Kelly et al. From 2014. And that means within this drop of water, you have information about the phytoplankton, bacteria, different organisms that have impacts on ocean and human health, zooplankton, and even for larger organisms that travel through that water, they might leave skin or fecal or tissue cells, something that's known as eDNA. So really the key for a lot of us is to access that molecular information that's captured within that particular droplet. And that gives us accessibility towards, what kind of biodiversity is present, how do organisms respond to their environment, are there things that are harmful or toxic, and then also a lot of things, or a lot of research is also geared towards looking for specific organisms that can be indicators of environmental change. And most of that requires that droplet of water or that sample of water be assayed by molecular methods. And traditionally, access to molecular information has really meant returning that sample to a shoreside laboratory for processing. And so what I'm going to really talk about today is how we can move those molecular methods onto aquatic robotic samplers. And that means kind of three big things. If you're operating in the field, you have to know how to collect a sample, and that sample has to be representative of what you're trying to process for. The method that we've utilized is particle concentration, mainly by filtration. And then you have to determine steps that are required for hands-off fluidic manipulations, and those include things that, and in different methods like ELISA for, say, the detection of toxin, or qPCR for maybe the detections of genes and transcripts. And it's not just the steps that are required for that, but it's also handling it in ways that aren't normal. For example, if we're going to deploy this, there's no refrigerators, and so things like reagent stability is really important. You can't always grab a clean epitube, and so you're reusing a lot of these fluidic paths. And so it also means being able to clean up properly between samples. And in a lot of oceanographic research, I'm going to sort of talk a little bit about some of the issues. You're not necessarily dealing with just plastics. You might have to utilize different types of metals, which may or may not be compatible with some of the reagents that you have. And so the lab provides a really benign environment to do research. It doesn't change very much in temperature, and you have a lot of access to things. But as soon as you go into the field, all of these new things kind of crop up. And so I'm going to talk a little bit about some of the challenges of working in the ocean. And for those that work in the field, some of these challenges they'll be very familiar with. One of those is power. How do you operate your instruments? And when you're in the lab, you have a really great source of 120 volts that's almost continuous, and you can have as many instruments as you want plugged into that outlet. But as soon as you go out into the field, it means operating on batteries. So most of the time we're operating on a limited amount of volts. 14 volts is what we're really pulling from. We also deal with a lot of different elements. We're out in the UV if you're in the upper water column. We also have to deal with saline water, and that's really a big impact on what kinds of metals you utilize to build your instrument out of, because you have to face then metal corrosion. pH and temperature may also fluctuate. One of the biggest ones that we deal with in the ocean environment is pressure. And if you look at the top of the screen, there's a video of a styrofoam cup shrinking as it's being carried to deeper and deeper depths. And what's happening is the air is being squeezed out of that particular cup. And if you look at our instrument, here's an example of a diver, again, hanging out near our ESP. And if you think about what pressure is doing on that can and our instrument inside, it's like at about 33 feet below the ocean surface, we already have one elephant standing on the lid of that can. If we drop down to 100 meters, we've got 5.5 elephants standing on that can. So pressure is a really big thing that we have to overcome as we try and access deeper depths in the ocean. And lastly, biology and biofouling is a big deal. So there's the can on the right that just went in. It's really clean and pristine. And 1 month later, you can see how many things have decided to take up residence on our particular can. So biofouling is a big deal because it can change the weight of your instrument. Things like birds or seals can perch on different buoys, and your instrument could even be attacked by a shark. And so our solution to a lot of these issues initially was the second generation environmental sample processor, or ESP. And this particular instrument, it's an electrochemical device. You'll see bags, and those are our reagents that are get accessed by various valves. We operate in a currency called a puck. And these particular pucks, we can load different kinds of filters in them, and they get loaded into this carousel. And what I'm going to do is show you a video as to how this instrument performs underwater. And you'll see this surface expression is at the top. This particular instrument is moored. It's hanging below that surface expression. And what's going to happen is that video is going to cut away, and it's going to show you how that particular instrument works. And so the first thing that's going to happen is that we're going to load a puck into a collection position, and we're going to make a fluidic connection with a clamp so that we can access seawater. Seawater is going to be drawn in across a filter. Organisms are going to get concentrated on that filter. We'll then add a chaotropic agent to lyse those particular cells open to make the DNA, RNA, proteins all available. We can collect that lysate or homogenate, then load another puck that contains an array of organisms that we're interested in detecting. We can add that lysate we generated. It can bind to spots on that array. We then wash it away and add reagents to develop that array. And then at the very end, we're going to take a photograph, and that'll tell us what kinds of organisms were present in the water. So this is a detection for ribosomal RNA, and in this case, it's for bacterioplankton. And the best part of this is that all of that data gets beamed back to shore within about an hour and a half of a sample being collected. So some of the lessons that we learned from deploying these second generation ESPs was that being at a moored location wasn't actually always the best. And a moored location means a fixed location. And so in 2015, we used both fixed and mobile assets in order to track a harmful algal bloom that occurred in 2015. And what this plot shows is data from our mobile asset. This is this autonomous underwater vehicle that was able to provide information about the water column. So here's depth from 10 meters down to 40 meters as that vehicle moved offshore. And what it did is measure chlorophyll in the water. And here you can see a tongue of high chlorophyll water. That's this red band that stretches from near shore to offshore. And what you also see is the star where our instrument was deployed. And it was collecting real-time data as to toxins that were present in the water. But as you can see, we're missing the peak of the bloom. In addition, we also learned a lot of lessons about our chemistry. And one of those is that we were utilizing custom reagents. So you couldn't buy those commercially, and they weren't necessarily in standard use in many laboratories. In addition, we had to use multiple extraction reagents in order to get measurements of toxin as well as the presence of the algal species. And that meant taking more samples just to determine the presence of those two analytes. And in addition, many of our automation steps, it required just way too many steps in order to analyze a sample. And one of those examples is in this automating of qPCR. So we'd have to collect a sample, lyse it with guanidinium, and then run a column purification that was multiple wash steps, and then elute it in water before you could even utilize that DNA in a qPCR reaction. And lastly, we would have to then, when we're finished, clean up and reuse all those fluidic paths, and we noticed that sometimes we were really prone to contamination. So that information really led us to two solutions. One is we really wanted to keep it simple, keep the chemistry simple, and to have a platform that allows us to go mobile. And so this is a design overview of our third generation ESP. We're much smaller than that second generation ESP. So we went from about the size of a large garbage can, a large garbage can, down to about the size of an instrument of say 2 basketballs. Okay. We also want to preserve the ability to sample and preserve those samples to bring it home for laboratory analysis, but we also want to make sure that we can create a homogenate and then analyze that and provide data still in real time. And a lot of the analytical modules that we're targeting for downstream, again, is for toxin and also nucleic acid detection. And so right now we're working with a surface plasmon resonance where we're utilizing that to detect toxins via a competitive ELISA method. And then eventually we're trying to work out how to design that analytical module for both PCR and for sequencing. And so all of our upstream processing has to support those particular methods. And what we came up with is this idea of a cartridge. A cartridge is basically going to handle all the sampling and all the processing that needs to happen upstream of handling that off to an analytical module. And this is just sort of the comparison between the two instruments. So remember the ESP, we have all these common reagents. We have that carousel of pucks, and it's this large instrument that then supports running all of those samples that can be housed in it. So now we're moving on to this cartridge. It fits into your hand, and it's everything to collect and process one sample. So a very, very different way to lay things out. This cartridge then gets plugged into our instrument. This is the third generation instrument portrayed here. These cartridges get loaded into the carousel. This is where they make a fluidic connection to the seawater, and so you have ability to concentrate particles on a filter. There's also this reagent side to the cartridge that interacts with different actuators that we have here, and we're able to move fluid in and around and off cartridge. And the presence of this minion down here, well, that's where this analytical module is going to sit when we deploy in the water column. And this is an example of a full ring of cartridges loaded into our carousel. We're getting ready to deploy this particular instrument. A cartridge that's selected that runs this lyse and go process is one that we're actually trying to analyze the sample in real time. And so the steps that are involved in that mean that we need to collect and concentrate particulates in the water column. We're going to evacuate that sample water. We're going to add a lysis buffer and heat. We have to be able to recover that extract. And then we're going to push that extract off cartridge for analysis. And that could involve dilution with different types of reagents. And so that real-time analysis is really dependent on sample homogenization. This is an example of our lyse and go cartridge. It requires heat so that there's some sort of additional features to this cartridge. And what we're really going for is a single lytic agent for all target molecules. And this means something that's compatible with both toxins and also for nucleic acids. We also want to know that the reagent, our lytic reagent, is stable at room temperature for a really long time. Okay, we're not going to operate in a refrigerator. We also want to minimize the number of steps that we have to do before we deliver to the analytical module, so we don't want to process very much. And so what we really did is start looking into the field and looking at different companies that were developing different lysis methods. And we ran into this company, MicroGEM, that produced RNAGEM and prepGEM. And they offered a solution where there's a cell lysis and no purification. And so this sounded really good to us. So what we started doing is trying to look to see whether or not RNAGEM will meet our needs. And these next set of experiments we did in a laboratory in a really controlled setting and in Eppendorf tubes and in sort of normal laboratory tools. And we asked 3 different questions. One, whether or not it lyses our targets, and that means it has to meet or beat our current methods of extraction that we utilized on our second generation ESP. The buffer system also needed to be compatible with our target molecules. For us, again, that means nucleic acids and some low molecular weight toxins. And most importantly, it had to be stable at room temperature over a long period of time. And so we were trying to shoot for at least a month because of how we were operating with our mobile asset. And this means 2 different things. One is the reagent stable at its working concentration. And then ideally, the extract that it produces would also be stable so that whenever we got back after deployment, we could actually still utilize that extract that was produced. This first slide shows a comparison of the lysis methods, and we were looking at nucleic acids. And so we were comparing different methods of extraction. One is that we compared RNAGEM to a QIAGEN all-prep method that utilizes bead beating. And then secondly, we compared it against what we utilized for our second generation ESP, which is basically a modified DNA-easy column extraction. We used 12 different seawater samples that were acquired through different seasons and at different depths to get a really wide range of different organisms that we might target. And so on the x-axis is these different genes that we targeted in the bacterial plankton. Those range from bacteria, also archaea, and then a diatom or a phytoplankton. And what this graph really shows is the log difference between RNAGEM minus that from the other extraction method. And anything above 0 basically means that RNAGEM won out. And so in comparison to our second generation method, it's kind of a no-brainer. The RNAGEM definitely outperformed. As far as it versus a method that utilizes bead beating, there were some groups where it was better with the RNAGEM and other groups where it was worse. One of those, Synechococcus cbbl, is known to be a hard to lyse sample, so that one wasn't surprising that it actually worked a little bit better with the bead beating method. Then we looked at the stability of the enzyme over time, and we stored this enzyme in working concentration, so it was in a 1x aliquot, and we stored it in room temperature in the dark. And then we looked at a shallow water sample and a deep water sample, and we assayed for those same groups. So we looked at the presence of total bacterial 16S, an archaeal species, and also the diatom. And we looked at this over a wide range of time. So we went anywhere from T0 all the way up to 334 days. And what we found is that enzyme was stable for really long periods of time. And there really was no significant differences between any of the time points that we collected. And so this was a really great find for us because it meant that we weren't just limited to just 1 month or a few week deployments. We could actually be out with this particular lysis buffer for really extended periods of time. We also then looked at the stability of the extracted nucleic acids over time, and really what this does is provide an option to utilize that extracted sample post-recovery. And so if we know that the extract hasn't changed, that it's representative of whenever we made that molecular measurement, this is a really good asset for us because we can confirm or perform additional assays. And so what we did is we reanalyzed an aliquot of the T0 extract that was held that at room temperature over time, and then analyzed that with qPCR. And we utilized the same targets. So we have the surface and the deep water sample. This is the log gene of abundance, and this is over time. So black is T0, 24 hours is in red, 13 days is in green, and you'll see that we didn't have any significant differences after 13 days. And then we analyzed at 49 days. We had one replicate from day 49 in the surface sample that was significantly different. But regardless, this is really promising results for us. So if we can get our cartridges working, then at least we know practically the sample that gets extracted is representative utilizing this particular buffer system. We then started looking at what happens to lysis onboard 3G. We're utilizing this in a different way. It's different materials. We're handling it very differently. And so what we're trying to do now is compare our 3G ESP process to the bench lysis. And so there's two different experiments that happen every time we utilize one of these instruments in the field. We do a pre-deployment positive, and in this case, the RNAGEM has been loaded in the cartridge and then added to the instrument. It usually takes us about 48 hours in order to set up this particular experiment. We then run our pre-deployment positive. We do 3 runs on the bench with the same water as we use on the ESP, and this is before it's deployed. So that tells us when we get it back and we analyze the extract, it tells us how long our extract is stable. And what we found is a little bit more variation within the ESP collected sample, but those samples were representative of what we collected on the bench. We then can also do another post-deployment positive, and that tells us information on how long the RNAGEM was stable in our cartridges. And in this case, it was 14 days was the duration of this particular deployment. And we found that the instrument that went into the field with those cartridges and that was stored for 14 days were just as good as RNAGEM out of the fridge. And so those were our bench controls. And so this really shows that at least operationally, what we're doing is working, and the samples that we're collecting are representative of what's going are representative of how we're collecting samples in the water column. So everything that I've discussed so far has shown that RNAGEM is compatible with looking at the various nucleic acid targets we're interested in. But I also want to talk about some toxin analysis because that's also really key. We really want to try and do this real-time toxin detection. And initially, we started looking to target domoic acid, and it's a neurotoxin from marine algae. It's a common issue in Monterey Bay. We have harmful algal blooms that generally occur each year. The method that we're going to utilize to detect is a competitive ELISA using an SBR. And here's the 3G instrument with this analytical module, the embedded SPR within it. And traditionally, the extraction method that we've utilized on our second generation ESP was 50% aqueous methanol with 0.1% tween 20. And we're going to compare that with RNAGEM to see if it's compatible, because if it is, then we're able to basically get to collect one sample and be able to get toxin and gene measurements out of a single extract. In order to see if this is going to work, we have to actually compare two different things. We have to know if that buffer is compatible with running SPR. And the second thing is to know that if we run the extraction, if we're able to extract toxin. So the first slide is showing that RNAGEM is compatible with competitive ELISA. So again, that target was domoic acid. We extract and compared the two different, we generated standard curves with both aqueous methanol and RNAGEM. And the curves are located on your right. The top is the one for the methanol curve. And what's really important here is the dynamic range of this particular curve. And so if you look at the dynamic range of methanol and compare that in the lower red plot of that for the RNAGEM, you see that that dynamic range is a little bit different, but it's not grossly different. And so this was really promising for us in order to proceed into actually testing to see if RNAGEM is effective for extracting the toxin. And this was work, again, done by Greg Doucette, and this is some of his unpublished data. So in this graph, it's showing you the yield of domoic acid from a culture, and this is utilizing this methanol tween method that we utilize on the second generation ESP. It's incubated at 60 degrees. He also did a control where he increased the temperature to 75, and then we compared that to extraction using RNAGEM. And what you'll see is that the yields might be a little bit different, but they're still in a very comparable manner, and it's very, very doable. And so this was acceptable results. And so now we're able to basically we can use the same extract to look for the presence of a toxin as well as to look for the presence of the organism. In addition to detecting these marine algae, we also found that RNAGEM was effective at looking for other low molecular weight algal toxins, including ones found in freshwater environments. And to that end, many of you will be familiar with a harmful algal bloom that happened in 2014 that shut down, closed the Toledo water supply. And so about 500,000 people were unable to drink their water. They couldn't cook or bathe for almost 3 days because it was contaminated by a toxin produced by a harmful algal species in Lake Erie. And one of the reasons for that is Toledo's water supply is actually located right here. And this particular area of Lake Erie is very, very prone to harmful algal blooms. And a lot of these are fueled by agricultural runoff. So here is the water intake. That's the water crib of where Toledo gets its water. And you'll see around this particular crib, you see floating on the surface are these marine algae, these toxins, this toxic marine algae that's present. In 2019, we traveled to Lake Erie and brought our instrument in order to try and map out where these toxins were present. So here's a picture of our instrument getting ready to go in the vehicle, and here's the vehicle flying in Lake Erie. And you can see the bright green is where these cyanobacteria are located, showing you sort of the density and the extent of it. And if you look in the map, this is overlaid with a satellite image that shows the extent of the bloom in Lake Erie. And the more red the color, the higher the concentration of that particular algae is. And so the stations that are listed on this are stations that NOAA GLERL, they visit and they monitor over time. And the red track that you see is where we flew one of our vehicles to sort of look at the extent of the bloom. And we were able to collect samples along the way. And those samples are being analyzed right now by our group and also the group at GLERL to try and gain a picture of what the extent of that particular cyanobacterial bloom was in 2019. So far I've mainly spoken about underwater applications of our instrument, but there are many other aquatic environments that one might want to sample where launching in long-range AUV isn't practical. For example, traveling to rivers or streams. And so our solution was to repackage our instrument into a more portable device, something that someone could carry into the field, and that it could also be used as a means to assess water quality. And it's known that many water quality markers are harder to lyse. It generally means you're trying to open gram-positive organisms, and so harsher methods are required. In the EPA, they use a mechanical bead beating method, but that method isn't practical on our cartridges. So what we wanted to do is explore the use of two different products, one RNAGEM and a second one prepGEM, to see if enzymatic lysis can actually open up these gram-positives. And we want to do this on our system, on our portable system, in order to show that we can do this. So what we're doing is we're targeting these gram-positive Enterococcus that were added to spiked seawater samples. We did cultures to show the total number of Enterococci cells that were filtered in our samples, and then we also ran extraction tests. And so the EPA method is in the green bars at the far right, and this is the Enterococci, the cell equivalents that were filtered in that sample. So this is what was recovered utilizing the EPA beading method that's in the green bars. These orange bars here show results utilizing the RNAGEM method. And so you can see that significant numbers of the Enterococci in that sample were not effectively lysed by RNAGEM. However, utilizing the buffer system and the enzyme of prepGEM, we find that we actually beat the lysis of the EPA method. And this is all within the context of our cartridge. So now we're really poised as to being able to take this portable device out into the field and then start looking for basically fecal indicators and assess conditions of potential human health impacts. Most of the data that I've presented so far has shown that we can collect representative samples and process them in a way that's compatible with results to what we've obtained in a laboratory setting. On this last part, I want to showcase what we envision as the strength of this technology, this idea of a mobile genetic robot. And I want to start with a real example of ocean sampling that we performed a couple years ago in collaboration with the University of Hawaii and the Schmidt Ocean Institute. And we traveled off of Hawaii about 100 miles into the Central Pacific Ocean, and we launched one of our vehicles to study a really special layer known as the deep chlorophyll max. And these deep chlorophyll are higher in biomass than in the other parts of the water column. And they're unique because they're in a special place where these organisms get nutrients from below, and they're also in the layer where they can still have sunlight to photosynthesize. So you get these areas of higher biomass about 100 or so meters below the surface of the ocean. So the goal of this particular experiment was to fly one of our vehicles inside this deep chlorophyll max to acquire it, and then to drift in it over time. We then used a second vehicle, OPA, to basically provide the environmental context, so it would fly around the vehicle and provide water column information above and below as Aku, the vehicle that was in the layer, state. This was really important because if you leave the layer, it's hard to get back in the same location you were. So we were utilizing two robots, one to do the sampling, the other to provide the contextual data. We also wanted to stay underwater as long as possible, so we also used a third robot, a wave glider, in order to maintain the position of those particular vehicles. You can see we were also way away on the ship, and we were able to keep satellite communications, so we can track the instruments. We can also perform other experiments and then come back whenever we decided we wanted to. So the goal of this experiment was to acquire that deep chlorophyll max, to remain in that feature, and to archive samples. And that's what Aku's job was. This is the sampling long-range AUV. We also, again, used OPA to profile around that long-range AUV up and down to provide the contextual information. So this video that I'm going to show you is a 4-day, 3-night drift of the sampling AUV, while the profiling AUV is going up and down in the water column and providing the context. So in this feature, there's going to be a black line that shows you the trajectory of the sampling AUV, and the colors are going to be the contextual data that was reported by the profiling AUV. The left panel is in temperature, and the left panel is in temperature, and the right panel is in chlorophyll. And the more green this area is, the higher the chlorophyll. So what you're going to see is this black line. This is the sampling AUV. You can see that it's sticking in this particular green chlorophyll. It's staying at that temperature, and it's acquiring samples over time, and it's able to stay and remain in that layer. What you might also notice is how much that layer moves around. It's moving at least by 10 to 20 meters per day night cycle. And so we were able to maintain and stay in that particular feature and drift with it over time. So the Hawaii example shows the ability of the long-range AUV to find and remain in a particular layer. And we envision that the real-time molecular data will also inform when and where to sample. For example, if you wanted to really wait till you saw a big bloom, you might have deployed before where you don't have a very high chlorophyll signal, and the organisms, say, in this quadrant, aren't present, and so you don't want to waste samples. So you're going to fly the AUV around and collect samples every once in a while until all of a sudden you reach a high bloom condition where you see high chlorophyll, and those organisms that you're really interested in studying are present. And then they're going to kick off and collect extra samples in those particular areas. And so this is how we envision having an intelligent sample scheme. And our ability to react to a situation really depends on how quickly we can get that molecular real-time data. And figuring out what reagents and methods that support that goal has really been an important component to the development of our ESP. I want to acknowledge members of the ESP team, and that's in the upper picture, as well as members of the ESP long-range AUV team, and that's the lower picture. Some of the work that I also presented was work that we did with the University of Hawaii, including with Ed DeLong and Dave Carl, and then our collaborators from NOAA, Greg Doucette, Reagan Era, Steve Ruberg, Paul Denull, and Kelly Goodwin. And the majority of this work was funded by the David and Lucille Packard Foundation, the Betty and Gordon Moore Foundation, and a few NSF grants to Chris Schulman. Thank you very much for listening, and I'll take your questions now. Oh, I can't see anything.
And thank you, Christina, for that informative presentation. And we'll start the live Q&A portion of the webinar now. So if you have any questions you'd like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. So let's take a look at our questions already coming in. Christina, our first question is, what is the salt concentration of the concentrated sample, and do you have downstream issues with qPCRs? If so, do you make any additions to your PCR mixes to improve the simplification?
Well, thanks, Susie, for that question. Whenever we try and sample, we're concentrating seawater, which has a salinity of about 35 parts per thousand. But the particles are getting concentrated, not the salt on the filter. And so one of the methods that we utilize to get rid of the sample itself is we evacuate all the seawater out. And so when we add the buffer, we have less than somewhere between 100 to 50 microliters of residual seawater left. So it's not making a big impact on our sample. Sometimes we do have inhibition that comes in with the sample, and we've overcome that by just doing a simple dilution. So usually a 1 to 5 or a 1 to 10 dilution is enough for us to get past the inhibition, and that usually has to do more with the organisms themselves than the seawater.
And how can we ensure that the sampling process itself does not disrupt the marine environment?
Yes, that's a great question. MBARI, we sit too in a marine sanctuary, so we are we are much more restricted with what we can do in the bay, and we're a lot more careful here. Everything that we deploy, we have to recover. So all the equipment we have to bring back includes recovering anchors that we utilize to hold our instruments in place. We also don't release any chemicals. So anything that what we do release is the filtered seawater, but we don't release any other chemicals whenever we are sampling. We have waste containers inside of our instruments that hold all of that material. So it's really the structure that might impact organisms within the marine environment, but we, again, after we're done sampling, we remove that from the environment.
Thank you, Christina. And I want to thank our audience. These are great questions coming in. We have quite a bit. We'll try to get to as many as we can. This next one is a two-part question. What are the advantages of conducting research in laboratories at sea? That's the first part. And what kind of underwater research can produce results more effectively than research that can be done on land? In particular, what kind of research can be done underwater that are not directly related to marine life?
Okay, those are really great questions. First of all, it has to do with when you really want that information. For us, if you're thinking about monitoring or even like a management situation, or you want to find an environment where you can target organisms that you're really interested in, it's nice to be able to have that information before you start sampling rather than after you go to all this really big effort. In terms of things that are more amendable, obviously the less steps and things that are easy to automate are much simpler. Things that take a lot of processing or a lot of steps or require a lot of informatics to interpret results are a little bit harder. So we found that things that are much faster, almost like a point of care where they give you an answer that you can then react to, have been the best. There are definitely methods that are not amendable to working underwater or at sea. And in those cases, things like where we can archive samples, or in the case where the enzyme is able to, whatever we lyse or the homogenate that we get back, if that's stable and we're able to analyze that further when it gets back to shore, those are all things that are really important to supporting this kind of research. And that's been kind of a key to us developing this instrumentation, is to know what we bring back into the lab with the instrument, or what we do at sea actually matches what normal laboratory methods have provided over the years.
Thank you, Christina.
Oh, and then the last question was the organisms that aren't, that aren't directly related to marine life. This type of technology you can also utilize to monitor for human health targets. So there's obviously large groups that manage human pathogens that they assess water quality, and they close or shut down beaches. And this type of research or instrument can actually be utilized to give more information in a faster, more real time. And so it would just be working towards qualifying this particular type of instrument for that kind of a management resource. And so it's really about trying to provide information about the environment in real time.
Thank you, Christina. This next audience member has a question about contamination. Mm-hmm. Does working in oceans increase the chance of contamination?
It definitely does. In that one of the pictures that we had showed earlier, there was a lot of biofouling. So it really depends on where you're at. So at MBARI, sometimes we operate during phytoplankton blooms or in coastal areas, and those instruments do get biofouled. We try to utilize like copper screens and things like that that might minimize different types of contamination. We also have ways that we can internally clean our tubes to try and our sampling tubes in order to keep down the, different types of biofouling, but that's definitely an issue in the environment. And we try and look at targets too that may or may not come in on that particular we try to target organisms that aren't necessarily associated with biofouling, but that's one way that we do that.
Thank you. And how long do robotic samplers last?
That's a great question. So a lot of times we are at the mercy of how much power we have. And so like the mobile device, that particular one will last about 3 weeks out at sea. The mooring that we showed you, that one has operated on its own battery pack for about 6 months. And then we've also operated, there's a cabled observatory that's present in the Monterey Bay, and we've been able to plug in that, and we were plugged in and underwater at 800 meters for about 8 months. So a lot of times it really has to do with your power. It also has to do with how many samples you want to take during that particular period. So if you want to try and take a sample a day and you have an carousel for the mobile device, there's 60 samples. So there you're really limited to say 60 days if you could be out that long. And so it really depends on how frequently you want to sample, where you want to be, how often your phenomena occurs. So those are all questions that whenever we lay out a deployment, those are all things that we have to answer.
Thank you, Christina. And again, I want to thank our live audience for their participation. Great questions coming in. Any questions not answered today will be answered via email from our presenter. Our next question, could you talk a little bit about how this remote sampling and analysis has impacted your resource allocations? I'm wondering about savings in terms of transporting the water samples, reduced lab plastics, or others.
Well, you definitely get really good samples. It impacts because you don't have a human that has to go out and collect those samples and filter them and then bring them back to the lab. The added benefit of those Lyse and Go cartridges is that all of that lysis is done while the vehicle's out, and so you're getting homogenates back that can be directly analyzed. So it's really just then recovering that off of the cartridge. Our cartridges right now we reuse them, and so we clean them and then reuse them again, and so there's less plastic waste in that realm, and then we're not using as many tubes and things like that, and that's been really the biggest benefit of that particular cartridge and that method. So it's not just the real time, but it's also the improvement on processing whenever you get back to the lab.
Thank you for that. This next question actually piques my interest too. Has anyone thought of using this in the testing of COVID, deploying to non-urban areas for testing?
You certainly could. It is a water sampler. It can be utilized and it can take on any type of water. It would really be, right now we don't have the downstream PCR requirements instrument in order to do that analytical provide that information in real time. But that's definitely something that the instrument can do. Those are targets that it can handle. And if that were something that somebody wanted to try, it would be possible.
Now, Christina, is all your molecular research limited to PCR amplification studies, or do they progress to amplicon sequencing of, say, the 16SR DNA to understand community representation?
So we definitely want to try and move into sequencing. We've already started. One of the other groups that we're collaborating with at MBARI has started to utilize the lysates that we generate on those cartridges to see how they represent, how different groups represent. They're mostly targeting larger zooplankton, so going after more eDNA products. And so that's definitely a vein that we're going down. A lot of the initial analysis that we did was to utilize qPCR because it's a quicker turnaround and it's easier for us to identify issues. And since some of the sequencing technology relies on a targeted PCR approach, if we can get the PCR to work and it's good enough and it's representative of what we normally see in the lab, we feel like it's a good transition to get to sequencing.
Thank you very much. Now, how often do you analyze the marine water?
How often do I, excuse me?
How often do you analyze the marine water? The marine,
so a lot of times what we're doing in the lab is a lot of development, but we try and do a number of deployments per year. In the Monterey Bay, we have between 2 and 3 big expeditions where we target particular events. So we've done things like studied diel migration of a zooplankton and looked at that acoustically and then targeted samples in high acoustic layers. We have an upcoming deployment where we're going to look at canyon dynamics, and so the distribution of different types of fish and bacterial plankton and harmful algal blooms around the bay. We also generally do 1 to 2 deployments outside of our area, and so the example that I gave in Lake Erie, we've been there for the last couple years, sort of utilizing this instrument in a different environment. So we're working in more shallow waters, fresh water, and for a group that does more monitoring.
Thank you for that. Now, what are the ill effects of the algae bloom?
Excuse me, what are the ill effects of the algae blooms?
Yes.
So it depends on the type of toxin. So the harmful algal blooms that occur in Monterey Bay, they're the cause of a diatom called Pseudonychia australis. And here the toxin that's produced is called domoic acid. And that particular toxin is a neurotoxin. So it has a huge effect on a lot of the seals and birds in this area. And that particular toxin, it bioaccumulates. So the diatoms produce it, then the anchovies or sardines eat it, that toxin gets accumulated in their body. So any marine organisms that eat that go through this, they suffer this neurotoxin, and it causes them like an amnesic, and they lose their sense, and it affects their brain and their hippocampus. And it is reversible if it caught early, and that can also cause issues to human health as well, and has caused the closing of numerous fisheries here up and down the coast of the U.S. on the West Coast. For example, it was the Dungeness crab was closed in 2015 because of the domoic acid bloom. In Lake Erie, the toxic cyanobacterial bloom occurs there and affects the Toledo water supply. That same cyanobacteria is also present in like the St. Johns River along in the coast of Florida, and those particular cyanobacteria can get to really high levels, and that's toxic to anyone that takes their pets to the beach or interacts with that kind of water.
Thank you for that. Now, could you extrapolate how your technology has been used or could be used for different environmental applications, for example, crop or pest management?
So a lot of this is trying to figure out ways to provide information in real time, and really our instrument is really just a sampler. It's good at sampling water, and that what we really tried to target was to make that our instrument very modular, so that anybody that wanted to develop an assay or add their own buffer, as long as they could operate within that structure of a cartridge, then that can work. We also try to make it modular in that any kind of analytical module could be plugged in. So we would provide a power and a fluidic and a comm link to it. And so we would be able to adapt different types of technology. So we're not stuck on just one. So as something new comes open or there's a new application, then we can figure out how to wrap our heads around what steps have to be done in order to meet those requirements, and that's really how we've progressed even into working like into the freshwater environment, because you can imagine, you know, those algal cells or the types of samples that they want to take being more near surface operate really differently in shallow water than how you would operate here in the ocean. And it, you know, the sample matrix is different, the types of organisms are different, and each of those questions mean changing either the chemistry or sometimes even the fluidic motions on the cartridges in order to make sure that we accurately represent information within that sample.
Thank you, Christina. We have time for a couple more questions. Do you use normal PCR or the QT PCR?
So we've been using quantitative PCR because we really wanted to get more than just presence absence. So we've done abundance. So in that second generation instrument, we worked with the Lawrence Livermore National Laboratory. They had a qPCR module that they had built for had developed for anthrax detection. And so we had utilized that inside of our instrument, and we built the PCR reactions on a and then would push those into that module. And so that was our mechanism of testing. When we move into the 3G, we're really trying to look at still trying to do something that will provide quantitative information, and whether that's via like a digital PCR or another type of quantitative PCR where you're dealing with larger slugs of fluid, both of those are really still on the table, and it really depends on what works best with our system.
Thank you, Christina. This last question, can you tell us about your first experience in this field, how you felt working for the first time, and if there are any common problems that you face in your work?
So I'm a microbiologist, and so this is a really fun project, and it's really exciting being able to watch and see work using robotics in the field. You really get to be out there a much longer time frame. You also get to watch, like in that video that we showed at the very end, showing us floating inside of a bloom. You really get to watch things change and alter. So it's this amazing experience of watching things transform in front of your eyes, and it's really that sense of discovery, of learning something about the ocean just as it's happening in real time at that moment.
Absolutely. Christina, thank you for your presentation today. Would you like to provide any closing remarks for our audience before we go today?
I just want to thank LabRoots and MicroGEM for this opportunity of sharing some of the work that we do at MBARI. It was a really a great experience for all of us.
And thank you, Christina, for your time and clearly important research. I'd also like to thank LabRoots and our sponsor, MicroGEM, for underwriting today's educational webcast. Please go and visit microgembio.com to learn more about rapid single-tube extraction workflows. You can visit them at their site. Before we go, I'd like to thank the audience for joining us today and for their interesting questions we did not have time for today, and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. LabRoots will alert you via email when this webinar is available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, be safe and take care. Bye-bye.